Filtration is a separation process used to separate solid particles from a fluid, liquid or gas, by passing the fluid through a porous medium, which retains the solid particles while allowing the fluid to pass through. In the pharmaceutical industry, filtration plays a crucial role in various processes, ensuring the removal of impurities, particulates, microorganisms, and other contaminants from pharmaceutical products and manufacturing processes. Common filtration techniques. 1. Microfiltration, MF. Description: microfiltration employs membranes with pore sizes typically ranging from 0.1 to 10 micrometers. It is used to remove suspended solids, bacteria, and large colloidal particles from liquids. Applications: clarification of pharmaceutical solutions, removal of bacteria and particulates from water used in pharmaceutical manufacturing, sterile filtration of liquids such as buffers, media, and drug formulations. 2. Ultrafiltration, UV. Description: ultrafiltration utilizes membranes with smaller pore sizes compared to microfiltration, typically ranging from 1 to 100 nanometers. It is used for separating solutes based on molecular size and is effective in removing macromolecules, viruses, and endotoxins. Applications: concentration and purification of proteins, enzymes, and peptides, removal of viruses and endotoxins from pharmaceutical solutions, recovery and purification of biological products such as vaccines and antibodies. 3. Nanofiltration, NF. Description: nanofiltration employs membranes with even smaller pore sizes, typically ranging from 1 to 10 nanometers. It is used to separate ions and small molecules based on size and charge, effectively removing divalent ions and organic molecules. Applications: purification of water for pharmaceutical use, particularly in the removal of salts and organic impurities. Fractionation of pharmaceutical formulations to separate active ingredients from excipients. Removal of endotoxins and pyrogens from injectable pharmaceuticals. 4. Reverse osmosis, RO. Description: reverse osmosis uses semi-permeable membranes to remove dissolved solutes and ions from water by applying pressure higher than the osmotic pressure. Applications: purification of water for pharmaceutical manufacturing processes, including formulation, cleaning, and rinsing. Production of water for injection, WFI, used in parenteral formulations. Removal of ions and impurities from process water and wastewater streams. 5. Depth filtration. Description: depth filtration utilizes porous media such as filter pads, cartridges, or diatomaceous earth to trap solid particles throughout the depth of the filter medium. Applications: clarification and prefiltration of liquid pharmaceutical formulations. Removal of particulates, microbial contaminants, aggregates from biopharmaceutical solutions. Filtration of cell culture media and fermentation broths in biopharmaceutical production. 6. Sterile filtration. Description: sterile filtration employs membranes with pore sizes small enough to retain microorganisms, typically ranging from 0.1 to 0.22 micrometers. It is used to ensure the sterility of pharmaceutical products and processes. Applications: sterilization of pharmaceutical solutions, including injectable formulations, ophthalmic solutions, biologics. Filtration of air and gases in cleanroom environments to maintain sterility during aseptic processing. Protection of downstream processing equipment and bioreactors from microbial contamination. Applications of filtration in pharmaceutical manufacturing. Pre-filtration of raw materials, removing impurities from starting materials like solvents and buffers before further processing. Clarification of intermediate solutions, taking out unwanted particles after reactions or crystallization steps. Sterile filtration of final products, ensuring the sterility of injectable drugs, ophthalmic solutions, and other sterile products. API purification, removing impurities and unwanted byproducts from the isolated active pharmaceutical ingredient. Filtration of fermentation broths, separating cells or biomass from the desired product in biopharmaceutical processes. Choosing the right filtration technique. The selection of the most suitable filtration technique depends on several factors, including stage of manufacturing, whether it's pre-filtration, clarification, sterile filtration, etc. Particle nature, the size, shape, and chemical properties of the particles to be removed. Desired product characteristics, the required clarity, sterility, and API concentration of the final product. Product compatibility, ensuring the chemical compatibility of the filter media with the drug solution. Economic considerations, cost of filters, processing time, and waste disposal. Various types of filtration. 1. Gravity filtration involves passing a mixture through a filter medium under the influence of gravity. The filtrate passes through the filter medium, leaving behind the solid residue, called the residue or retentate. Commonly used in laboratory settings for separating solids from liquids or gases. 2. Vacuum filtration, similar to gravity filtration but employs a vacuum to speed up the filtration process. The reduced pressure created by the vacuum increases the filtration rate by pulling the liquid through the filter medium more quickly. Often used for larger volumes or when a faster filtration rate is required. 3. Pressure filtration involves applying pressure to the mixture to force it through a filter medium. The pressure differential increases the filtration rate, particularly for viscous liquids or when high purity filtrates are required. Commonly used in industrial processes such as chemical manufacturing and wastewater treatment. 4. Buchner filtration, a type of vacuum filtration that uses a Buchner funnel, a porous funnel fitted with filter paper. The vacuum is applied to the flask below the funnel, pulling the filtrate through the filter paper while retaining the solid residue. Widely used in laboratories for separating solids from liquids, particularly in organic precipitate. Various types of filtration continue. 5. Centrifugal filtration involves spinning the mixture at high speeds in a centrifuge to separate the components based on their densities. The denser components move to the bottom of the centrifuge tube while the lighter components remain at the top. Often used for separating particulate matter from suspensions or for isolating cellular components in biological samples. 6. Membrane filtration utilizes a semipermeable membrane to separate particles based on size and molecular weight. The filtrate passes through the membrane while particles larger than the membrane's pore size are retained. Commonly used for sterilizing liquids, concentrating solutions, and separating colloidal suspensions. 7. Cross-flow filtration involves passing the mixture parallel to the surface of a membrane, creating a tangential flow. This prevents the accumulation of particles on the membrane surface, improving filtration efficiency. Used for continuous filtration processes, such as ultrafiltration and microfiltration, in various industries including food and beverage, biotechnology, and wastewater treatment. 8. Dead-end filtration, also known as surface filtration, involves passing the mixture perpendicular to the filter medium. Particles are retained on the surface of the filter medium, forming a filter cake, while the filtrate passes through. Widely used in laboratory and industrial settings for separating solids from liquids or gases. These filtration techniques are versatile and widely used across various industries for a range of applications including purification, separation, concentration, and sterilization. The choice of filtration technique depends on factors such as the properties of the mixture, the desired purity of the filtrate, and the scale of the operation. Disclaimer: this channel does not promote or encourage any illegal activities, and all contents provided by this channel are meant for educational purposes only. Any copyrighted material on these slides is included as fair use for educational purposes only. It will be removed at the request of copyright owners.